Hello, everyone. It's my honor today to introduce Jean Mulcahy-Levy. She is a valued colleague. She did her original degree in a medical school out at Portland, residency in Arizona, and she's been with us ever since she did her fellowship in pediatric hematology oncology. During her fellowship, she had the inspired choice of Andrew Thorburns' lab, and really her science has concentrated on autophagy in pediatric brain tumors since. She's come up through the scientific fire, initially with foundation grants, and then with a K-12, then a K-08, and now has an R01, which will actually start at the beginning of next month. She's won a fair number of awards on the way, and has actually presented at national and international meetings. And she's now an assistant professor in pediatrics and pharmacology and has put in for promotion as well. Above all, I think she's an inspiring and collaborative colleague. I would say that she is transformative on our group in pediatric brain tumors. And I think that she has the potential to actually lead our field. So without more ado, I will give you Jean.
Thank you very much, Nick, for that very lovely response. So I'm going to give a slightly different talk than just a straight all my research all the time kind of presentation today. And the reason that I wanted to do that is because one of the things that I think a lot of people on this campus that are doing research is their ultimate goal, right, is to think about translational research. And what are we doing that's going to transform how we treat patients, how we treat families, and advance care. So what I'm going to focus on really is harnessing the power of available drugs to advance the care of pediatric brain tumor patients. And so in 2016, what happened was that brain cancer, brain tumors became the leading cause of cancer death in kids, which had previously been led by leukemia. And so part of this is because leukemia has had such great success in their research in developing new therapies, and we have been lacking a little bit in our advancements in treating certain types of brain tumors. And so if you just search for the number of FDA-approved drugs that are specifically indicated for brain tumors, this is the list that you come up with. If you actually go through this list and you cross off all the ones that are repeats, what you really have is 7 drugs that are approved by the FDA to treat brain tumors. And we obviously clinically use a vast greater number than just 7 drugs and more than just these 7, but it's pretty indicative that we're lacking in specific drugs shown to work in brain tumors. And so really what a lot of people focus on is new targets. What is driving these tumors? How do we attack those particular targets? And what else is out there? And so if you look at this, this is just a bar graph showing the number of approvals for what they call NMEs, or new molecular entities, by the FDA. So if you look at 2018, there were 59 new molecular entities approved by the FDA, which is pretty good when you compare it to what they'd been doing previously when the drug approvals were only in their teens. But when you look at this, a lot of these NMEs are adult only. There's only one pediatric drug that was approved in 2018. It's not entirely an NME because it's really just a reformulation in something we call asparaginase, which is already used to treat leukemia. So really the advancement in pediatric-directed therapies is lacking overall, and in brain tumors specifically. So if you think about producing a new molecular entity, right? Like, what are we going to do to define new drugs? And so this is really a timeline for how that would be. And so if you see here, this is like your classic drug discovery. You take these 5,000 to 10,000 compounds, you do a bunch of in silico screening, you do a bunch of in lab screening, and you bring that down to maybe 250 molecules that then you do preclinical work on. So that's what we're all doing, right? All of our in vitro studies, all of our mouse studies, and all that kind of stuff. So then that is a low estimate, takes 3 to 6 years. So then you get all that preclinical data and then you take it into a phase 1 trial. And so if you look at your phase 1 trials, you get about 5 drugs out of your original 10,000 that you can take to a clinical trial, then you have to run them through a clinical trial. And so if you're not a clinical person, I'll just explain these steps really quickly. So a phase 1 trial, it's a small number of patients. These are typically safety trials. Is this drug even safe to give to humans? We've done all the toxicity studies in our mice and our dogs or whatever we used, but can you give it to a person and have it be safe to give? It doesn't necessarily mean that you're trying to find out if the drug is effective, just can I give it to someone and not hurt them? Once you decide that and define what a tolerable dose is, you move into a phase 2 trial. Phase 2 trials, right, so phase 1 you could do it between 20 and 100 patients, phase 2 100 to 500 patients. That's really your trial, like, does this work? Is this something that we think is going to be effective, and move it forward into further clinical trials or larger clinical trials? Then we have what we call phase 3 trials. This is what we think about as being like the big trials, thousands of patients in the adult world. I can tell you not in the pediatric world, but in the adult world, thousands of patients can be involved in those trials. And if you look at that, that's somewhere between 6 and 7 years in theory to get those trials done. I would say that is a very low estimate of the amount of time it takes to get things through a phase 3 trial. And then you actually have to monitor those patients. So you may get all the patients treated in 6 to 7 years, but you have to monitor them for up to 5 years before that data actually gets published. And then from your 5 to 10,000 compounds, really you've come up with one possible drug that eventually gets FDA approval, which itself takes a couple of years to do. And then once it's in clinical use, you have to follow it up indefinitely with reports. So that's a particularly long time frame to produce just a brand new drug. And if you look at the number of drugs that enter clinical trials, 9 out of 10 drugs that enter phase 1 clinical trials will actually fail at some point during their trial. So about 10% of drugs are discontinued in phase 1. They're found to be too toxic or you can't find a safe dose. 50 to 60% are discontinued in phase 2, so it's safe to give, but it doesn't really help or do anything. And then 20 to 35% are discontinued in phase 3 for who knows why. So if you think about it, this is not just time, but it's money. So if you think about, right, it's going to take you 15 years from drug identification to delivery, how much is that going to cost you? So there's estimates that it's somewhere between $2 to $2.7 billion and 8 to 12 years to get a drug from when you discover it to when you're actually going to get it FDA approved and on the market. And so if we all had this happy little guy in our lab who was handing us stacks of cash that we didn't have to apply for, then that would be fantastic, right? And so unless one of us wins the lottery and wants to fund all of us for the next several years, we have to consider not only the time we're spending here on the weekends and at night and early in the morning, but all the money we're spending to try and define where these new drugs are coming from. And then if you look at it, they did a study that found that there's a number of reasons for why these fail, and they estimate that $28.2 billion on U.S. spending is wasted because of research that is irreproducible. So if you want to think about that, so 10.8% laboratory protocol errors. That's where we submit our errors, and then these get carried forward into these phase 1, phase 2, phase 3 trials, and why the focus on designing our trials well and making sure that our preclinical data is good is important. But it's equally as important when we even get the drugs into patients, because we can have unreliable biologic reagents, improper study design, and then really inadequate data analysis and reporting, which is a big deal. Remember I said you can do a clinical trial in 6 years, but then you've got 300 authors and they all have to agree on the manuscript, and then you have to get it through revisions. And so all of these take time, all of this takes money. So that's a very down view on drug development, right? But not everything is so negative. There have been some very significant successes over time. And so one of the major successes that we've seen in the world of leukemia is this development of Gleevec, or these BCR-ABL inhibitors. And, this came out of Brian Drucker's lab when he was at OHSU. And he was I worked in the lab next door to him when he was developing this. They were fascinating when they were doing this. But if you think about it, this is a drug that was designed to do something very simple, in theory, is block this site to prevent growth of leukemia cells that had that mutation. And this drug was transformative in the care of patients in CML. So this is what it was originally designed for. So if you look at this, this is your growth curve. So this is survival time in months, and this is the proportion surviving. So the higher up you are, the more you've got patients surviving, and the longer out they're surviving. So if you look at sort of all of these other therapies, you can have other therapies, you can have stem cell transplant therapies getting you to maybe a 40 to 50% survival rate. When imatinib came out onto the market, or Gleevec came out onto the market, this is what it did for survival. It took you from a 50% survival to close to a 95% survival for a pretty significant amount of time. And that drug has now been expanded into a number of other diseases. So starting with CML, starting in adults, transferring over to other diseases that carry the BCR-ABL mutation and moving forward with that. A more recent study that's come out was this study from the New England Journal of Medicine looking at TRK fusion positive cancers and using litrectinib or NTRK inhibitors in the treatment of cancers. And so this is really an important paper because it is very rare that you see the words in adults and children in the title of the same manuscript. So oftentimes what we're doing is we're looking at developing drugs and we're saying, okay, well, this is a drug for adults or this is a drug for children. And so this really was a study that looked at more the fusion than anything else. It didn't matter your age, it didn't matter your diagnosis, and it really had disease and site agnostic responses. And so basically what that means is if you look at this, this is a waterfall plot. And so this line here is it's 0. If it's above the line, that means the tumor grew while it was on therapy. If it's below that line, it means the tumor shrunk on therapy. If it's below the dotted line, it means that it shrunk what we consider clinically relevant amount. And so in this case, they're counting 30% as the clinically relevant amount. And so then if you look at this waterfall plot, the majority of patients responded to the drug in some way. The tumor got smaller. If you look over here, the different colors, each bar is a different color because it depends on where the tumor was. So you've got breast cancer here in these orange bars, you've got thyroid in these light green bars. So there's site and disease agnostic. It didn't matter what the tumor was called. It didn't matter where the tumor was. If you had this fusion, the medication appeared to work, and importantly, it worked in adults and kids. So sometimes that $2.7 billion to develop these drugs results in a fantastic response. But this is the timeline of development for NTRK inhibitors. So I want you to note that it started back in the 1950s, and it has an approved drug in 2017 or 2018. And so, the work that people have done in the past and built up to design these drugs is amazing. And building on all of this knowledge that we have, not just from the most recent years, but, what do we have out there from the past that maybe we're overlooking or forgetting that will give us something we can use today? So it's this idea of repurposing or recycling drugs. So taking a drug that we already have, that's already had all of the money spent on it, and seeing, are any of those drugs going to be helpful in the diseases we're trying to treat today? And so when you look at repurposing in the field of oncology, which is what we're particularly interested here at the Cancer Center, really what you want to think about is if we take all these things that we work on in oncology, we work on resistance, we work on genome stuff, we work on microenvironment, and if we design drugs just for oncology, single drugs at a time, that's just going to be increasing costs. If we look at what we can repurpose or what other drugs can we use that we already have available, can we design some more sustainable costs, some more sustainable treatment for our patients? One of the new drugs that was approved in 2018 by the FDA is a new drug that's to treat osteoporosis, which has a $20,000 per month tag. And that is considered inexpensive compared to the other osteoporosis drugs that are out there. So, understandable, if they're spending $2.7 million to produce a new drug, then they should be in some way be able to recoup that. But what if we could take another drug that's been available and is no longer costing us $20,000 a month and get equal results or better results? It's also going to be able to shorten the timeline. So remember I showed you that timeline in the beginning about how long it takes to produce an NME or a new molecular entity. So that would be represented by this top bar here. If you look at the lower bar, what you can see is that we can significantly shorten that timeline because a lot of that pre-work has already been done. A lot of that defining the structure, defining what it attaches to, all of those things have already been done for us. And so we can use this information to get drugs to clinic faster. So if you just do your 4-square plot to think about how we can do this, we've got, rate of return, low or high, risk, high or low. And so really if you want high risk, high reward, right? So those are your NMEs. And those are very important things that we need to keep doing. But you can also get really high return with fairly low risk with the repurposing. And remember I said that you have, maybe 1 out of 10 drugs that make it through clinical trials. You have a much higher success, 25%, which isn't fantastic, but higher with repurposing. So with this in mind, I'm going to present to you just sort of an array of stories. Like I said, we're not going to go into the nitty-gritty details of some of the science, but if you have questions, feel free to stop me. But just really to give you sort of an overview as to what we are doing here on campus within the Pediatric CNS group to develop new therapies for patients. And so obviously you all work here. I can't imagine a single one of you has ever been to has made it this far without hearing about autophagy, but in case that has happened to you, what we have here is this process of autophagy. And so what it is, it's a multi-step process where you get some kind of signal that comes into the cell, tells the cell I'm starving or I'm stressed. That signal comes down into the cell, tells the cell to build this double membraned vesicle, which we call the autophagosome. That autophagosome chews up and takes up things within the cell, proteins, mitochondria, damaged DNA, and then that fuses with the lysosome here, and then it degrades the product within that, and that allows it to spit that stuff back out to the cell and gives the cell energy and proteins and things that it needs to survive, particularly under stress. The important thing that interested me was that there's multiple ways that you can inhibit this. You can inhibit it with ALK1 inhibition, you can inhibit it with ATG4 inhibitors, VPS34 inhibitors, but down here, what you have is inhibition using drugs called chloroquine and hydroxychloroquine. So they're thought to work in autophagy by blocking this fusion of the lysosome to the autophagosome. And so this is important because these drugs are old, cheap drugs. So chloroquine was discovered in 1934. It was originally considered to be too toxic for humans, and so it was basically put on the back shelf. And then in the 1940s, the military gave money to do research for treating of certain diseases, and they found that chloroquine was effective in treating and preventing preventing malaria. And so its friend and buddy in the chloroquine family is hydroxychloroquine. So this is a drug also known as Plaquenil. This was approved in 1955. And so importantly, this is also a drug that is on the World Health Organization list of essential medicines. And so if you're not familiar with that, the WHO list of essential medicines are lists that countries say are essential for the health and care of their population, meaning that if this drug were to leave circulation, that they would be not able to take care of a significant portion of their population. And so in 2016, 4 million prescriptions were written for hydroxychloroquine in the United States, which I think is fascinating because I'd never thought about it much until I started studying autophagy. Hydroxychloroquine, under the name Plaquenil, is being used to treat things like rheumatoid arthritis, porphyria, Q fever, as well as malaria and lupus. And more importantly, for my particular interest is it is used to treat kids. And so because we have a drug that's been approved for a long time, it's got information and data on toxicities and PKs and PDs and all that kind of stuff in kids, this would be a fascinating drug if we could find out that it worked for something in brain tumors. And so this study came out shortly as I was entering the lab back in 2009. And so what this is a study of adults with glioblastoma. It was a study done out of Mexico. And so what they found was that they took a control group. So this is your standard sort of survival curve that you see for GBM. It's a really horrible survival curve. And what they found was if all you did was add chloroquine to patients receiving radiation plus chemotherapy, standard Temodar, you change their median survival from 11 months to 24 months, which is a significant increase in survival for a patient population that has just horrible outcomes. And so we wanted to see, in kids, we have limited population, and so could we define kids who would be best targeted with this autophagy inhibition therapy? And so autophagy is very complicated, and this is just one figure to look at it, but one of the things that we figured out over the last decades of research in autophagy is that there do seem to be some markers of autophagy dependence. And so things like the P53 signaling, PA3 kinase signaling, RAS signaling, JAK-STAT signaling, all of these different altered pathways can be used as ways to say, well, this cell is more dependent on autophagy than a cell that doesn't have this. And this is really exciting in the world of pediatric brain tumors because there's something called BRAF mutations. And so BRAF mutations are something that constitutively activate BRAF. So if you look under the normal pathway, what you see is you've got receptor tyrosine kinases that signal through RAS. That gives a nice growth signal through something called BRAF, which will then go to MEK, to ERK, which then moves to your nucleus and tells a cell to grow. And if you're controlled by your tyrosine kinases in a nice linear fashion, you can have nice controlled growth. If you have a single point mutation in BRAF where you change at site 600 from, typically you'll hear it referred to as a V600E mutation, what happens is you lose these upstream regulators. You lose your RAS, you lose your RTKs, and this is going to be signaling all on its own, therefore causing uncontrolled growth in tumors. And so we know that this increased tumor growth, was discovered and defined in adult melanoma and colon cancers, but we also began finding this in CNS tumors, and not just one kind of CNS pediatric tumor, but multiple. So pleomorphic xanthoastrocytomas, gangliogliomas, malignant astrocytomas, or epithelioid GBMs we can see it in. And so we also began finding that this is a negative prognostic marker. So if you look at this graph on this side, what you can see is that if you're BRAF wild type, that's your black line on the top, you have okay survival. If you have a V600E mutation, that dropped your survival significantly. If you look at the other chart over here, what we did is here locally, we looked at our BRAF expression or BRAF status in our brainstem ganglioglioma tumors. So these are tumors that you can't remove by surgery because they're in the brainstem. They're tumors that don't respond really well to radiation or chemotherapy. And what we found is that if you had wild type V600E, so this dotted light up here, that although your survival was not awesome at 50% survival, if you carried the V600E mutation, we had 0 patients survive that particular disease. Some other data has come out recently. We're comparing sort of larger numbers of patient panels. And so if you look at it, this is a BRAF V600E and low-grade glioma. So your wild type patients are in red and your V600E patients are in blue. And you can see under here that it's got a pretty significant effect on your progression-free survival, as well as your overall survival there in panel B. And, we now have this mutation that we can see in pediatric tumors, and here's something super exciting, is that it's in melanoma. So the American Cancer Society predicts about 96,000 new diagnoses of melanoma in 2019. Of these, about 7,000 people are predicted to die in just 2019. So 50% of those patients diagnosed, or half of those 96,000 patients that are diagnosed in 2019 are going to be found to be BRAF V600E positive. That is a gigantic number of people. Compare that to, this is the estimated number of pediatric brain tumors from age 0 to 14 per year. So 3,720. So if this were a mutation that were only defined in pediatric brain tumors, there is not a single drug company on this planet that would spend $2.7 billion to create a drug that targets that. But because we have half of 96,000 adults every year with this mutation, that is a very potentially profitable drug pool, unfortunately, for the adults. So Thankfully, a couple of, not just one company, but multiple drug companies have developed drugs against this, vemurafenib, dabrafenib being the first generation drugs that target the V600E. And so what you can see here is this is, again, one of those waterfall plots. And so this is looking at the response of low-grade glioma to the response of low-grade glioma to therapy. And so these are all low-grade gliomas in general. And so again, above this line, you have kids that grew on therapy. Below that line, they're responding, and below the red line, they're responding in what we consider a clinically significant manner. And so what you can see is that patients in the blue and the green that were treated with standard of care being radiation, chemotherapy, or some combination of the two, that really it's sort of half and half, that these patients are growing or responding, and really not a large portion of them are making it below this red line. This list of patients here on the end, these are patients that were identified with the V600E mutation and treated with either vemurafenib or dabrafenib. So every patient treated with the drug had a clinically significant response. And so this is one way that we're sort of taking this benefit of the adult V600E inhibitor and starting to translate this down into pediatrics. So now we'll circle back to autophagy, right? So one of the things that we can look at is how much autophagy does a cell have? How important is it to the cell? And we can find that if you look at cells that carry, brain tumor cells that carry the V600E mutation, that they have much more autophagy under stress. And so basically what you're seeing here is under standard media, these cells have basically the same amount of autophagy. That's great. But if you stress these cells, what you can see here is the dark bars compared to the white bar, where the white bar is your wild type, is that the cells with V600E are inducing much more autophagy in response to stress, giving them better survival advantage. And then if you inhibit autophagy important proteins like ATG5 and ATG12 compared to your wild type here in the white, what that shows you is that these cells are exquisitely sensitive to loss of this particular pathway. So the question becomes, right, are two repurposed drugs better than one? So here you've got your chloroquine, here you've got your vemurafenib. So can we combine these to make it even better therapy? And so what we did is your standard sort of in vitro curves. The top line in here, or the the dotted line on these graphs is drugs that were treated with whatever drug is listed below. So you've got vemurafenib, which is your targeted. Your middle one is cisplatin and bemblastine on the end. And then your top two rows are cells that had the V600E mutation, and then your bottom row is a wild-type BRAF tumor line. And what you can see is that in all the V600E lines, regardless of whether or not you're using a targeted therapy or you're using a standard chemotherapy like cisplatin or bemblastine, that blocking autophagy improved cell death in these cells. You can develop resistance to these targeted therapies. So this is just showing you that if you give the drug over a period of time, that a stronger, hardier, happier tumor cell is going to develop. And that if you treat these cells with a combination of your targeted therapy, so vemurafenib plus your autophagy inhibitor chloroquine, you can effectively reduce the cell growth again. So, you can make these cells die better with a standard chemotherapy. You can make them die better if you treat them with a targeted therapy. And if you become resistant to those targeted therapies, you can make that targeted therapy work even better. You can use something like autophagy, which is more like a global cell process as opposed to a singular signaling pathway. You can actually use that to overcome multiple forms of resistance. So in this case, we've got cells treated with media, the single drug, the inhibitor, or the autophagy inhibitor, and then your last row of each of these is your combination, and the bottom one is different mutations. And so you've got NRAS mutations and KRAS mutations. You can improve therapy. You can improve response. The blue line there at the bottom is your response when you have EGFR overexpression, which is a known resistance mechanism to these targeted therapies in brain tumors. You can take cells that would never respond to your targeted therapy, so cells that, say, carry a P10 mutation. So these are cells that, if you look here, these cells don't really care. They've never seen targeted therapy before, but they carry a P10 mutation, so you could treat them with V600E inhibitor all day long, and these cells are going to keep growing. But if you combine it with chloroquine, you can see here how much a better response that you get. So you could potentially make a drug available to patients who would never be able to use it in the clinic. So that's great. I didn't show any mouse data. This is all in vitro, fantastic, who cares? Only 1% of drugs actually make it clinically, no matter how good my bar graphs are. So do these results translate to clinic? And so this is just one example of a patient that we've treated here who had previously been treated on B600E inhibitors. And so what you can see here is look at the red bar. And so the red arrow points to a part of the tumor. So for people who don't read MRIs every day, this would be pretending like the patient's standing up, the top of their head, the bottom of their head, and that bright white thing is the tumor. And so this patient, if you treat her with vemurafenib or a V600E inhibitor, you can see that that big white blob got bigger, right? So we treated her with a targeted therapy, and her tumor actually grew. So then the thought was, well, maybe we'll treat her with something downstream. Maybe like a BCR-ABL inhibitor, maybe it's developed a mutation within that pocket. This drug can no longer fit. We'll treat her with something downstream so that we can still block the same pathway we know is driving the tumor, but we'll block it a different way. So that's what we did here, treating with trametinib, which is a MEK inhibitor. And so what you can see is that big white blob still got bigger. And then we decided what we're going to do is go backwards, and we're going to treat her with vemurafenib and chloroquine. And what you can see here is this big white blob started to get smaller again. And so that just shows you that, thankfully, all of this stuff I was really excited about, every bar graph I made, all my purple plates actually do translate into a clinical response. So if you think about deliberate modulation of autophagy, really, we think about a lot of trials that we do, and we don't really think about targeting them to specific populations, particularly in autophagy. It's sort of, if you have breast cancer, come here for your autophagy inhibitor. So the BAM trial, which is a BRAF autophagy and MEK inhibition in metastatic melanoma trial, was the first autophagy trial to really target a specific mutational group. And so in this case, looking at the V600E melanoma patients. And so my argument has been that we should, in pediatric CNS tumors, do our initial autophagy inhibitor studies in patients who have BRAF V600E, and particularly on patients that have either failed single drug BRAF inhibitor therapy or combination BRAF MEK inhibitor therapy. And so thankfully, this is the PBTC trial that's coming soon. So this is the Pediatric Brain Tumor Consortium, one of our early phase clinical trial in pediatrics. And what we've done is try to rationally design this trial to get the best positive outcome. And so what you have here is two arms. We have patients that will be molecularly characterized with either a V600E or a low-grade glioma, or if BRAF fusion if they're low-grade glioma. And then it'll be phase 1 just to define the dose of hydroxychloroquine for kids in these combinations, and then breaking them out into phase 2s. We'll be designing this trial in such a way that every patient can be their own control so that we really minimize the number of patients we need to be able to show a clinical response. So for example, we only need 6 high-grade glioma patients with the BRAF V600E mutation to be able to show enough clinical response to say this would be worth taking forward. So taking that hundreds of patients you would need in a phase 2 trial, designing your trial in such a way to maximize the data from every patient that you have, and taking that 100, 200, 300 patient need that you could get in adults but you can't get in kids, and move it down to somewhere between 6 and 12 patients per group. But what about other tumors, right? So one of the other tumors that we look at is something called ATRT. So this is sort of a historical survival curve. So at the top, which you can see, if it's hard for you to see the numbers, is basically that we were giving them a generous 25% chance of surviving 5 years. If they were metastatic, this would be this tiny little growth curve over here. And what we've done over time is really tried to intensify therapy. Can we just give them so much chemotherapy that we can overcome the tumor? And that's what we're doing now, is this high dose therapy. And that's improved us. It's gone from 25% to 42 and 53%. If you're less than 3 years of age, we tell our families how great we're doing, that we have it up to 39 to 45%. But these little guys here, this is one of my little guys with ATRT, I don't want to have to tell them that, I think we're doing fantastic. We can cure almost 45% of these kids. welcome to our treatment world. And so we really need to define new tumors and find ways where, the therapy that we're using is so toxic that sometimes these kids can get very sick just from what we do to them. So one of the things that I know a lot of people do on campus is drug screening. And so there's a lot of tools out there that you can use for drug screening. One of them is approved oncology drug sets that you can get from the FDA. They used to have 134, now they're 147. You can get them, they don't charge you. I think they're free. So if you want your very own set of FDA-approved oncology drugs, you can get nice little 96-well plates with them. And so what we did was, using this NCI-approved drug oncology panel, is we took multiple cell lines of ATRT and we did the panel. And so what I want to show you down here is that these, because you can read all the names, are drugs called bortezomib and carfilzomib. And so pretend this one's carfilzomib, this one's bortezomib. And so this is both of these cell lines. So this is in a VT12 and a VT16. These are sort of your old school ATRT cell lines. They've been around forever. This is in a 737 cell line. So this is a cell line patient-derived that we developed here. And you can see that our proteasome inhibitors, our bortezomib and carfilzomib, are on there. So that's really exciting, right? You do a drug screen, you find a hit that's really big, you go and you do your search of the previous data, and there's some data out there that, sure, they looked at bortezomib in solid tumor stuff in the past, but it doesn't really help me because that doesn't cross the blood-brain barrier. But what does cross the blood-brain barrier, in theory, some people will argue it does, some people will argue it doesn't, is a new drug called marizumab. So what you can do then is you can take your hits, and if it's not necessarily the perfect drug for your disease, is there something very similar that you can analyze? And so in our case, there was this drug called marizumab. And so this is looking at a number of cell lines. So you've got your VT12 and your 16s, your sort of grandfather ATRTs on the bottom. On the top, you have 3 patient-derived cell lines that we've developed here. And what you can see is that sort of in these long-term growth assays that really nanomolar doses down to the 10, 25 nanomolar dose is really quite effective in vitro. If you look at them and grow them as neurospheres, which is more in sort of their natural environment, you can see that the size of the neurosphere volume is significantly decreased, both in your patient-derived line on 737s as well as your BT12s and BT16s. And then if you take them in vivo, now this isn't the best data, and this is some early runs that we've had, but if you look at this over here in this patient-derived in vivo sample, this purple line is your one of your cohorts of mice that have ATRT given to the poor mouse, and then you treat them, and that you can show that they've got increased survival there. You can also then take these approved panels to try and do some work to see what you can do, right? There's the rare cancer that we treat with a single drug. And so we're always thinking about combinations. And so what we currently do is we take a new concept drug and we add it to an old backbone. We're going to take this drug, we're going to add it to what we're already doing, and that's how we're going to make our combination. And so one other way that you can decide what might be good combinations is to do sort of combination drug screens. And so what you have here is that our original drug screen on the top, and then here's our bortezomib inhibitors. And then what you have here on the bottom is that exact same drug screen done in the exact same cell line, but now in the presence of with or without marizumab, so 25 nanomolars of marizumab. And so all of the red dots that you're seeing is what that new drug response is in the presence of marizumab. And so what you can look at this is you could then go through and say, wow, these drugs up here used to make the tumor grow, but this drug, for some reason, when you give it with marizumab, all of a sudden you get a much better response to whatever that drug is. I can't remember what it is, and I can't read it, it's too small. Although Andrew, who does the work in my lab, probably knows what it is. But you can see here that you've got all of these potential combinations, right? These are all FDA approved. Maybe some of these drugs are already used in these kids. Maybe it would make sense to add it to the backbone that we treat ATRT with. Or maybe our relapsed ATRT kids could be using marizumab with something else, something that we wouldn't have considered putting together before for whatever reason. So, that is kind of an overview of some of the major projects in my lab. But I'm obviously not the only one here. We have 7 pediatric neuro-oncologists here. We have 2 people in our group Katie Doris and Lindsay Hoffman that are clinical trial specialists, and then the other 5 of us that run labs. And so what I wanted to then quickly do is show you a little bit of just a couple of highlights from some of the other labs. So out of the Vibacher lab, who in addition to doing this kind of work has numerous other sort of hardcore molecular-based things going on, really what can we look at that's moving rapidly to clinical trial? So back in 2012, Jeff Nipstein, who used to be a fellow here, did some work looking at the use of drugs, HDAC inhibition specifically in ATRT. And so there's a drug called entinostat, which is an HDAC inhibitor. It blocks HDAC 1 and 3, or class 1, class 3. And so what you're seeing here is mouse studies that look at, on the top you've got your control, and on the bottom you've got your entinostat mice. And so what they're able to show is that this drug that's already approved being used in adults can significantly reduce the ATRT growth in vitro. We can show it in our mice. We can show it that treating these mice gives us a smaller tumor volume in vivo. We can show that these mice live longer. And we know that this drug is potentially effective. So this drug, based on primarily this work, is now moving forward to clinical trial. So you can see from 2012 to 2019, which is really 7 years, which sounds like a long time, but if you compare that to really the 10, 15 plus years you need from a new drug, we're able to take something that was out there and available and now moving it into kids. This is some very elegant work that's coming out of Adam Green's lab, who's another one of our researchers. And so he's got a particular interest in treatment-induced high-grade glioma. So I mentioned to you earlier that a lot of the stuff that we do to kids causes them some harm. So radiating children at a small age, giving them high-dose chemotherapy at a small age puts them at risk of developing tumors in the future, and particularly treatment-induced high-grade glioma. So these high-grade gliomas are particularly devastating because although the glioblastoma graph I showed you of the adults, they had some maybe survival, there's no survivors of this tumor. These are very aggressive, nothing we have that works. And so what he's done is really a lot of work looking at how do you identify these tumors, can we classify these tumors, and can we take those classification of tumors and looking at these different, we have developed some cell lines here for them. You know, can we, looking at these different classes of medications, come up with things that could potentially be used to treat these patients? And so, for example, up here in this box, what you see is based on their classification of Group A and Group B tumors, you can get some different responses to different drugs like bortezomib, again, proteasome inhibition, and etoposide, or you can go by class of drug here to define which drugs are going to be more effective. And so this is looking for drugs we have right now to potentially treat a disease that we have absolutely no cures for. If you look at Dr. Foreman's lab, who so nicely introduced me, you can look at how are we going to treat ependymoma. So ependymoma has about a 60% 5-year progression-free survival, meaning only 60% of kids do not regrow their tumor within the first 5 years. And so what his lab did was an interesting screen where they didn't screen just ependymoma cells, but they screened ependymoma, ATRT, medulloblastoma, high-grade glioma in a way to compare them. So trying to define what, this is a little off. So what drugs were ependymoma specific? And so could we define something that would work specifically for ependymoma? So really this phrase should be up here. So what it should show you here is that your ependymoma cells are more responsive to drugs called 5-FU and Atra. So 5-FU is an old school drug used a lot to treat colon cancer, and Atra is a drug used to treat leukemia. So if you take those, you can treat them doing your in vivo models. So for people who don't read mouse MRIs all day long, this is a mouse brain. This is the cerebellum, so sort of the back of the mouse and front of the mouse. And then what you can see here is this bright stuff here in these upper ones. Those are your tumors. And so if you treat these tumors with vehicle, you can see they can get to be quite large. This tumor is compressing all the brain forward, backwards, and sort of squishing down. That's what ependymoma likes to do. And then if you treat them, what you can see here, for example, is this is the comparison of the tumor in a mouse that's treated with a 5-FU and Atra protocol with radiation. And so again, not drugs that you would specifically consider putting together before, but both available, FDA approved, and cheap in theory because they've been around for a long time. The Foreman lab is also, and I apologize that these titles have come down, but the other big thing that's sort of out there in the world today is this idea of immunotherapy. How can you harness the immune system? And so there are ways where you don't have to necessarily go out there and create the next greatest big CAR T cell that's going to treat cancer. There are things out there that you can use already. And so what they found here was that if you looked at your ependymoma cells, what they were able to find is that ERB-B2 and IGF-2 therapeutic monoclonal antibodies could be targeted in these tumors. And what you have is the potential of using drugs like trastuzumab, which targets your ERB-B2, and GM-CSF, which is a drug that stimulates your macrophages in the body. And so based on that previous data, the Foreman lab was able to open a clinical trial, and this is just an example of a clinical trial response of someone with relapsed ependymoma, this bright spot here, that was treated with this protocol using GM-CSF, where you give the GM-CSF for a couple days to rev up your immune system, then you give the antibody with the hope that this antibody will stick to the tumor, and then those revved up immune cells will target that particular cancer and kill it off, and then you can see that the tumor has essentially resolved. So this isn't just something that the Pediatric Brain Tumor Group is on. This is something that came in our email yesterday. If anyone wants to go to it tomorrow, but repurposing drugs and devices to accelerate biomedical innovation. So really, if you think about it, we need to continue doing our work in CRISPR screening. We need to continue our work in defining new molecules. We need to do our work in defining these new structures. All of that is very important, but while we are doing that, we can also be working on using what we already have. Save ourselves some money, save ourselves some time. So this work from my lab, so this is the people doing most of the work. So you have Mish, Shadi, and Andrew, who are fantastic and have really, Andrew did the ATRT work out of my lab that you saw. Shadi is responsible for the majority of the autophagy work that you saw. Mish is new to our lab, but don't you worry, you'll see something from her in the future. But really, all of the neuro-oncology labs that we have here on campus, and this is not the most updated slide, but you can just see the number of people that it takes to get this information quickly, and having our clinical research support from Katie and Lindsay is essential because those are the people that are going to take what we do in the lab and make it into a clinical trial, show that it works, and move things forward. So that's all I have today. If you have any questions, please let me know. Question?
Great talk. You showed an example where, for example, mice were getting treatment and the survival was beyond 100 days, and that tumor came from a patient with just wondering, sometimes you want these new mice, but these patient tumors, maybe the patient died and the mouse is still going, and it makes me wonder how much of the mouse studies actually are possible to progression of clinical trials because we don't potentially components, particularly the immune system.
Right, so PDX models, right, so patient-derived models are what really important. We collect tumor sample at diagnosis as well as relapse if we can, and so a lot of these models that end up working are the patients that have more aggressive tumors. For example, that ependymoma mouse model is from a really aggressive tumor. And you're right, it's in a not immune competent mouse. And so that is going to limit some of your potential translation into people. That's probably why a lot of the clinical trials or things that get to phase 1, only 1 out of those 10 drugs that get there end up going into actual production because maybe some of our data was really not relevant to humans. And so there's a lot of work out there about creating new mouse models, trying to do these tumors within immune competent model systems. I know the leukemia groups are really trying to use, mouse to mouse syngeneic kind of studies that they can do. We do the best we can with the tools that we have. And so that's why I think really using in vitro data, using primary patient tumor samples, using PDX models, using established models, I think if you can take all of that information together, that will hopefully lead you to something that's more relevant in an actual person instead of relying just on a single piece of data. But it's not always true, so you do have to be selective. Yeah?
You've shown that your BRAF inhibitor in combination with chloroquine works much better than cells.
Correct.
So you looked at the autophagic response to a single drug treatment, say with the BRAF inhibitor. Sometimes these inhibitors cause upregulation of the autophagic one. So that would be one of the reasons your combination works better than single drug treatment.
So if you look at vemurafenib or dabrafenib BRAF V600E inhibitors in melanoma cells, the answer is yes, that you actually do get an upregulation of autophagy in those cells. If you look at if you treat CNS tumor cells with a BRAF inhibitor, they are already so ramped in their autophagic response that it's really hard to detect a further ramping of autophagic response. And so I do believe that it's... Yeah, I believe that those cells are actually dependent, because if you inhibit, it's very hard to, so if you do like an shRNA knockdown of those cells, it's very hard to get them to grow long enough to get your results. So my lab is very thankful that every single time they do the experiment, they have to transduce a new set of cells. Like we can't just make a cell line and then like let it grow, because we're killing off so many of our cells with the inhibition. So it's really, I think, a synergistic effect how drug A is working and drug B is working, not just a response to the autophagy and RAF inhibition. Any other questions? All right, thank you all for coming.